as a candidate, because he's a associate professor at Duke and he'd risen through their own. So they were working like hand in hand in Echo, doing strain, the era of strain where it was new. We did some really good stuff together, but at that point I had a couple different roles, but
but I'll see him because
so he's one of the senior, so he's your attending.
Yeah.
Now, do you know what you're going? Are you going to have a subspecialty or
heart failure?
Okay.
But Mads, he moves into advanced heart failure.
So Mads did, okay.
Yeah, he's doing. So heart failure is going to be my
good. Okay. Yeah, and you and because you did some imaging stuff.
Dr. V, Rohan is going to introduce.
Are we sharing? Here goes. I think we're live. There's no Zoom counter today. Where the heck is it? Can you see us online? Can you hear and see us? Last control was pretty good. So that's all. That's it. Hi everyone. Welcome to another Grand Rounds. We will get started right because we're just a few minutes over, and I want to have enough time for questions at the end. Today we took the opportunity and the privilege of having ACC right around the corner to invite our distinguished speaker today. But before we get in, I just have a couple of quick announcements. First off, the CME code is 47792. I hope people can see that on the screen. We have a suite of talks lined up, including several visiting speakers from Northwestern and Weill Cornell, but also several fellow talks and discussions that are going to happen through May. This is the acknowledgement disclosure section as part of our HCME program, including for Dr. Bering Sorensen. So before I hand off the stage to Tor, my friend, don't let his young face make you feel like that he is early in his career stage. He's a full professor at the University of Copenhagen and has had a distinguished academic background with medical training at the University of Copenhagen, as well as advanced training in clinical sciences and discovery, both from University of Oxford and Harvard. He is the founding director of the CTCPR, which is Center for Translational Cardiology and Pragmatic RCTs, which is a collaboration between the University Hospital and the Biomedical Sciences Division, where he is the professor. This is a large group of 50 affiliated members who have led some of the most largest pragmatic clinical trials in Denmark, leveraging the registry systems in Denmark to conduct these trials into flu vaccines and other large initiatives, which I hope we'll learn more about how to think creatively within the construct of what we have and build these new discovery systems. So without further ado, I'm going to pass it off to Dr. Bering Sorensen. Thanks for being here, and thanks for sharing your vision with us.
Thank you. Thank you so much, Rohan. How did you move the slides? Was it there? Okay, perfect. Thank you so much. It's such a great honor to be here today, and it's already been an amazing day. I've seen the grounds of Yale and met some amazing people that have inspired myself and my career. So thank you again for the invitation. This is my disclosure, and I saw you already showed that disclosure slide, so I'm going to move on right away. So first of all, we can ask ourselves the question, why is it we need registries for the conduct of our randomized controlled trials? And due to the success of Eric and many of the other trialists that have conducted research within cardiovascular medicine over the last many decades, event rates are getting lower and lower every year. This is just a graph showing these event rates for cardiovascular mortality in the UK. And when we test a new treatment, we are almost always testing only for moderate treatment effects. And these two things combined, the fact that we're looking for moderate treatment effects and the low event rates, means that our trials need to get larger and larger, and larger trials also results in more cost and more complexity. So that is why registries potentially can help us to continue getting the highest level of evidence by embedding randomization. During the COVID pandemic, the Oxford group, they was quite concerned about the fact that many health authorities, they said we need to use real world evidence to figure out how we can overcome this problem of this new disease. And they wrote a commentary in New England Journal of Medicine with the title, "The Magic of Randomization versus the Myth of Real-World Evidence." And now I'm just quoting the authors from the Oxford group in this commentary. "Promotion of non-randomized analysis of databases as a rapid source of real-world evidence about the effects of treatments is a false solution to the problem caused by the bureaucratic burdens imposed on randomized trials," right? But maybe we can combine the two worlds, combine real-world evidence, real-world data with randomization, and thereby getting the best of two worlds. So first, I'm just going to talk about how registries potentially can improve the conduct of trials. And we can use registries to improve the trials in several ways. I'm just going to mention a few, and I'm going to hopefully, during the talk, you're going to see how we at least have tried to execute some of these things. We can use registries to identify liable patients for our trial. We can use registries for patient screening, for patient enrollment, enrolling patients directly at home. We can record patient consent through our registries. We can use registries to randomize the patients. We can obtain information about baseline characteristics, and of course, follow up both within the trial, the execution of the trial, but also when the trial is finalized. We can also follow up patients that didn't participate in our registries. It reduces the burden on site staff, but the next, then I'm going to mention the burden of participants, is probably the most important thing. That, not burden the participants of trials is probably the most important thing to the road of success. And I'm going to show you some example of this. We can use registries to enable novel trial designs, as I already mentioned, direct to participant trials, embed registry trials in our registries. We can do trials for lower cost, which means that we can do larger, sufficiently powered investigator-initiated trials, which is often the thing. We're limited to the big cost, which means that we need to do a compromise and then do a trial that isn't sufficiently powered because we just don't have enough money to do the realistic or the true sample size at which we need. Meaning we can do more trials because we can do them for lower costs. And I'm going to show you that we are doing several simultaneous trials because they're so cheap to execute. We can assess additional questions, additional subsidiary outcomes can be assessed, sick days away from work, a lot of other stuff that we're assessing in our trials. We can assess the long-term effects, as I already mentioned, and it can potentially improve the quality by having complete unbiased data on our trial participants. So this is just a schematic trying to explain for those of you who haven't worked with registries as much as we have back home. In general, we stratify our registries into two types. the administrative registries. That's, the national registries, the population registries, the claims databases, and the clinical registries. That's those where there's disease-specific clinical quality registries, clinical data registries. And the type of data that these two registries entail are also different. In the administrative registries, we have hospital admission, we have discharge codes, we have outpatient visit, diagnosis code. In the clinical registries, they are disease-specific, right? So it's disease-specific biomarkers, procedures. And if we look at the clinical registries, many of you probably know a big portion of the trials been conducted in these registries. It's the Sweetheart registry, for example, which have done many pragmatic trials, but it's a clinical registry. So the Swedish investigators that conduct trials in this registry, they still have to wait for the patients to be admitted for them to include them in many of their trials. That is very different from our trials, which we conduct in administrative nationwide registries. And I'm going to show the examples very soon. So when we talk about these administrative nationwide registries, the Danish registries are part of this group. And these three registries that I show here on this slide are just a list, 3 out of several other registries, but these 3 registries are just paramount for any kind of research question that we want to assess. They are the patient registries where we get information about inpatient and outpatient contact in our Danish free-for-all public healthcare system. Then we have our medicines registry. It's the prescription registry where we get all information about any prescription being prescribed in the country. And then we have our causes of death registry, which in its name tells what we obtained from that kind of registry. One unique thing that we have in Denmark, besides these registries, which actually exist in many countries, is a mandatory electronic email system called the e-box system. That is linked to the social security number that we can identify in our registries, and we can thereby contact everyone that we identify in our nationwide registries. And this specific thing, at least the legal approval of us contacting people through the administrative registries, is quite unique. And that's, I think, the agenda that we need to push many other places, that we should be allowed to identify people through administrative registries and contact them for potential participating in trial. But the e-box system, as mentioned, it's linked to the social security number. So we can identify every diabetic in the country and then contact them through this system. It's an email system that is only used for priority mailing. So it's only when you get your paycheck from the government, when you get information about your pension funds, and when it's time to get vaccinated. And it, of course, allows us for a rapid delivery of recruitment letter, but also trials where the letter is the only intervention that we're testing. So this combination of the nationwide registries in combination with the e-box system makes Denmark currently a great place to do large-scale randomized controlled trials. So this is just to show you the different trials that we are going to talk about today, the case examples I'm going to show you. First, I'm going to take you through the trials where we have used our nationwide registries in combination with the e-box system to identify, screen, and invite trial participants for trials that have been conducted with conventional logistics. So there we only use this system to boost recruitment. That's going to be the DCLOTE CKD trial that I presented yesterday as a late-breaking presentation at the ACC, and the Coherent trial. Then I'm going to take you through how we use our registries in combination with the e-box system to identify, screen, and invite trial participants for trials where all parts of the trial are being conducted and executed in using this system. So we are recruiting, we are randomizing, making it possible for us to do large-scale outcome trials, and that's going to be our flu vaccine trials, the DanFlu-1 and DanFlu-2 trial, our RSV vaccine trial, DAN-RSV, the DanLogic heart failure trial, and the DanNutri trial. Then I'm going to take you through some case examples where we use our nationwide registries in combination with the e-box system as the sole intervention. So the letters are the only interventions that we test. So we have we are doing implementation trials. I'm just going to show you our notch flu trials for this concept. And then I'm going to take you through lastly how we use the nationwide registries in combination with the e-box system to test the efficacy and safety related to time of delivery of chronically administered drugs. And that's the aspirin C3 trial. We're doing a lot of these trials, but I'm just going to show you the aspirin C3 trial. So first, it was the concept of using the registries in combination with e-box system to boost recruitment in conventionally conducted trial. The two case examples is the DCODE CKD trial and the Coherent trial. So the DCODE CKD trial, which, as I mentioned, I presented yesterday at ACC, we wanted to test how come SGLT2 inhibitors provide such cardiovascular protective effects in patients with chronic kidney disease. So it's a small, mechanistic trial, and we see the patients for echocardiography at baseline and after 6 months of treatment with dapagliflozin versus placebo. But we are a cardiology department. How will we identify 222 patients with chronic kidney disease without cardiovascular disease? That's quite hard, right? So there we leverage the registries to identify everyone in the vicinity of our hospital with a chronic kidney disease diagnosis code and invited them through the e-box system, making it possible for us to recruit 222 chronic kidney disease patients without cardiovascular disease in 1 year and finalizing this trial. Just to contrast a multinational trial that is being conducted in Denmark, the EASY Kidney trial, it's BI that is funding that trial. They plan to recruit 150 CKD patients in the whole country. We randomize more patients in 1 year at one center than they are planning to do for the whole country. The other example is the Coherent trial. And the reason why I'm mentioning this trial is a trial where we're testing colchicine as a potential antihypertensive drug in patients who have hypertension, but again, with no cardiovascular disease. We don't see them at Department of Cardiology because they're being treated at the DP. The diagnosis code in the registries for hypertension, it's also rubbish because, you only get a hypertension diagnosis code if you are sick with a lot of other stuff. And here we want to recruit a clean hypertension cohort, right? So there we went into the registries and identified people with hypertension based on them being prescribed at least 2 antihypertensive drugs and making sure that they don't have a diagnosis code of any cardiovascular disease. This trial, we also finalized recruitment, but we're still waiting for the last follow-up. The trial is conducted in conventional way, but there we managed to recruit the 150 pure hypertensive patients in a very short time span. So just 2 examples where we use 2 different registries, either the ICD-10 codes or the prescription registry to boost recruitment in conventionally conducted trials. Then we are moving on to the large-scale outcome trials where we use the registries to all parts of the trial conduction, also for the baseline characteristics and for the follow-up of events. So the first trial that I'm going to take you through is the DANFLU-1 trial, which we executed a couple of years ago and presented at ESC as a late-breaking presentation. It was a pilot trial. So the pilot part of it was just learning, does the ethical committee, does the Danish Medicines Agency allow us to do a trial conducted completely solely through the registries? We never see the patients on, besides when they go down and get the flu vaccine, and all safety surveillance is also conducted through the registries. So the pilot part was actually figuring out, is this possible? Is it allowable? Will we get through with the innovative ways that we wanted to do? And at the current moment, the DANFLU-1 trial was a pilot trial and a feasibility trial, as I mentioned. But currently, we have 2 flu vaccines. We have a high dose and a standard dose flu vaccine. And the phase 3 trial that Sanofi, they funded in 2015, just showed that the high dose flu vaccine lowers the risk of having a positive flu swab, right? But no vaccine trials have been powered for clinical outcomes. It's not like we do in cardiology where we need to power our trial for clinical outcomes. And the reason is they know that they would need to do a trial of 300,000. That's just not possible within the cost of conducting it with conventional trial features. So they need to embed a lot of pragmatic features if they want to do an outcome trial for vaccines. But the DanFlu-1 was just a feasibility trial testing whether it's possible. But because we were randomizing people, we went into the registries afterwards and saw whether there was any differences in these clinical outcomes in our feasibility trial. The way we did it was we identified candidates through the nationwide registries. We invited them through the e-box system, guiding them towards a homepage where they saw a video of me giving the informed consent for the trial. You know, all the information that they received was just from me through a video. They signed the informed consent with an electronic signature. We guided them towards the clinics, the vaccine clinics, which we collaborated with, where they then checked in with the social security number. They were randomized to a high dose or standard dose flu vaccine. And that was the only two data points that we received. It was the social security number and the randomization, what they were randomized to. That was then uploaded into the nationwide registries, where we got all data on baseline characteristics and follow-up data. Safety surveillance was done, so we were pinged every time. That's what's shown here in the middle, where my group is placed. We were pinged every time a person was hospitalized through the nationwide registries. And then we have access, nationwide access to the electronic health record. So we could, in a much more timely way, assess safety, because we could go in and look at the hospital charts as soon as the patient was admitted if one of our trial participants were admitted to hospital. And this is the reason why the Danish Health Authority said, okay, your safety surveillance in this kind of trials are much more timely than what we get from conventionally executed trials. So they were extremely happy. But this is just to show you the flow. We ended up in this pilot trial inviting 34,000 Danes. Of these, 12,500 were finally randomized to either high dose or standard dose flu vaccine. And that number in itself is also quite impressive, that more than 1/3 of those that we invite through an e-box go in and sign the informed consent and are randomized in our trials. And what I already mentioned in the introduction, the unique thing is that then you can compare your trial population, as you can see here, the DanFlu-1 population with everyone else in the country. I just want to see, okay, I can't figure out the pointer, but you can compare your trial population with everyone else in the country when you execute your trials within this system, really assessing the generalizability of your trial population and also assessing the healthy volunteer effect, right, of people participating in research. As you can see, we also saw that the DanFlu-1 population were healthier than the overall Danish population. But we did see some comorbidities, for example, I think it's asthma and cancer that was more prevalent in our study population. But we still see the healthy volunteer effect in these pragmatic trials. And it wasn't powered for clinical outcomes, but since we randomized these 12,500 citizens, we assessed whether there was any differences in hospitalization events. And as you can see, we saw that those who were randomized to the high-dose flu vaccine had a significantly lower risk of being hospitalized for influenza and pneumonia, corresponding to a relative risk reduction of close to 65 percentage points. That's, very, very high. And for most of the other hospitalization events, it went in the same direction. And very surprisingly, we also saw that those who were randomized to the high-dose flu vaccine had half the risk of dying of all causes. But again, these numbers only hypothesis generating. We didn't power our trial for these events. But the DanFlu-1 trial was such a success that we convinced Sanofi, who paid for the trial, to move on to do the sufficiently powered outcome trial, testing whether a high-dose flu vaccine significantly lowers the risk of being hospitalized for influenza and pneumonia. It's also co-powered for cardiovascular outcomes as compared to the standard dose flu vaccine. And we plan to randomize 340,000 over 3 seasons. This is the actual recruitment curve. As you can see on the right side, it's the total number of randomized individuals. And you can see we started the first year, 2022-23, with a small recruitment rate, but the 2 following seasons, and we managed to randomize 160,000 each year over a period of 4 weeks. So it's showing the numbers that we randomized per day. As you can see, the day we randomized most was a Monday in the 3rd week of the 2nd season, where we randomized more than 15,000 in one day. We have then gone on to, and I just want to say that this trial, we are hopefully going to be presenting at ESC, the main results, because we are going to follow them until 31st of May in the registries. And then we have clinical outcomes on all these 332 participants in the summertime. We also moved on to execute the Dan-RSV trial using exactly the same setup, but we were quite nervous because the RSV vaccine is a new vaccine. Everyone knows the flu vaccines, right? But this has only been approved a little more than 1 year. But we wanted to test whether RSV vaccine significantly lowers the risk of hospitalization compared to no RSV vaccine. And that's why we designed the Dan-RSV trial. We used the same concept, identifying everyone in the country above 60 years of age, sending out 1.3 million invites to these participants, guiding them towards a homepage where they saw the video again of me signing the informed consent. The only thing that differed was that they got randomized as soon as they signed the informed consent electronically to either show up at the vaccine clinics to get the RSV vaccine or to stay at home to not receive a vaccine. This design was due to the fact that Pfizer, who funded this trial, were not willing to pay for placebo. So we had to do it this way in a pragmatic sense. And of course, this design has some limitations with crossover. You know, people go down and purchasing the vaccine themselves if they don't get randomized to it. But again, this is just a recruitment rate, and you can see the actual dates of on the x-axis, and we beat the DanFlu-1 recruitment rate on 1 day in Dan-RSV. As you can see here on the 4th of December, we randomized more than 20,000 in 1 day. So it seems as if we can still beat our own trials in recruitment, and my parents are participating in all of these trials. Sorry, I can see something has happened with the slide. Moving on to something that is a little more interesting for us as cardiologists. I learned a couple of years about Boston Scientific pacemakers. Apparently have a heart failure alarm called HeartLogic. And this alarm has been part of all their ICDs since 2018. So it sounds as a great system, which measures impedance, of course, respiratory rate, as you can see on the right side, night heart rate, sleep incline, weight, activity level, and so on, and combines all of these measures into one measure called the HeartLogic Heart Failure Index. If this goes above a certain threshold, I believe it's of 12, the alarm starts. And Boston Scientific has already shown that if this alarm is elevated, your risk of being hospitalized for heart failure increases significantly within 30 days. And currently in Denmark, no one is paying to get access to this data. So we have a completely HeartLogic naive population where no one is using this information. It's just, you know, the alarm is just going on and no one is doing anything. So I suggested to Boston Scientific to do the DanLogic heart failure trial, again, going into the registries, identifying everyone in the country who have a Boston Scientific ICD device who is HeartLogic compatible, and then just randomizing them in the registries. Half of them, my group at Gentofte Hospital, are now calling up, asking, you know, if the alarm starts, initiating or intensifying diuretics to test whether these remote monitoring HeartLogic alarms and acting on them potentially can reduce the risk of heart failure hospitalization and all-cause death. So just another example. The last outcome trial example that I'm going to take you through is the neutral heart failure trial, because it's, of course, much easier to doing pragmatic trials when it's a vaccine. It's one jab, and then you just follow everyone through the registries. Getting an infrastructure up where you send out tablets through a prolonged period of time is much harder and much more difficult. So we designed the neutral trial where we are testing whether Q10 or selenium might improve outcomes in patients with heart failure in Denmark. The way that it works is that we're doing it in a 2x2 factorial trial design. But again, we identify all Danish citizens with a diagnosis code of heart failure. We invite them to participate in the trial through the e-box system again, guide them towards a video. And this time it's one of Eric's good friends, Lars Kober, who is the person in the video explaining the participants about the trial, because we believe that he was the most famous heart failure cardiologist in the country. So we thought it was better that he was the one explaining all of this. Then we sent out the drugs or the nutrients and the placebo through the postal service. So again, we are randomizing people at home. We are never seeing them. We are never in contact with them. And then we are just assessing the clinical outcome through the registries. The last theme that I'm going to take you through is where we use the e-box system and the letters as the only intervention. And that's our implementation trial to increase uptake of vaccines, but also our circadian rhythm trials that I'm going to take you through lastly. So we wrote about this in New England Journal of Evidence, just scoping of the concept of this, trying to nudge Danish citizens to live a healthier life. It can be whether they nudge them to get the vaccines. It can be whether nudging them to do more exercise, drink less alcohol. You can nudge people to a lot of things. And this concept where you come up with a idea, a hypothesis, you design your letters, and then you randomize every Danish citizen who fulfill the inclusion criteria to different letters, and then you just follow them through the registries. And we wrote about this in this concept paper. The first of our nudging trials was the nudge flu trial, which we published 2 years ago, where we want to test whether we can nudge Danish citizens to get vaccinated. The way that we did it was that we identified all Danish citizens 65 years and above. That's close to 1 million. And then we randomized them in a strange fashion, as you can see, 9 to 1 to 1 to 1, to either receive usual care, that's not receiving a letter from us, or receiving 9 different behavioral informed letters that smart behavioral scientists had helped us invent to see whether how we potentially could increase the uptake of vaccine. And this is just to show you the different letters that we tested. And then again, after 3 months, we assessed the primary outcome, whether we changed the uptake of flu vaccine. Then we followed them through in the registries through the whole season to see whether a potential change in vaccine uptake could be translated into differences in clinical outcomes. And that's also the unique thing, right? If a nudging strategy is positive, you can also assess what that means in clinical outcomes. For example, uptake of ACE2 inhibitors. Here it was flu vaccine, but you can do any kind of nudging trial. So this is just to show you the letters that we designed. So they were actually the same. The only thing that differs was this area where it says placeholder for text-based nudges. Otherwise, the letters were completely the same for all of the 9 different letter types. I'm not gonna take you through all 3 examples of the nudging sentences that we included in this letter. I'm just gonna focus on the top one, because that's the only nudging strategy we as cardiologists, invented ourselves. All the other nudging strategies was invented by or was put in or recommended by behavioral scientists. So we invented this new cardiovascular gain nudging sentence, where we wrote, in addition to its protection against influenza infection, influenza vaccination also seems to protect against cardiovascular disease, such as heart attack and heart failure. Here you can see the study flow. As mentioned, we randomized close to 1 million Danish citizens, and we had approximately 50,000 individuals in each of our intervention arms. We saw that 2 nudging strategies significantly increased vaccine uptake. And you have to bear in mind that this was on top of a baseline vaccine uptake in the usual care group of 80%. That's very, very high. Denmark and Portugal were the countries in the world with the highest flu vaccine uptake here in 2023. But 2 strategies were useful in increasing vaccine uptake. The first was just sending out a repeat letter. So where you didn't enter a notching sentence, but just reminding. That makes sense, right? Reminding people of now it's time to get the flu vaccine. It increased the vaccine uptake with 0.73 percentage points. But the single most effective notching strategy was the only one that we as cardiologists came up with ourselves, the cardiovascular gain letter. It increased vaccine uptake with 0.89 percentage point. So that's a clap on the back that we can actually, invent stuff, notching strategy that is more successful. We also assessed whether our cardiovascular gain letter had a differential effect depending on the baseline characteristics. And as you can see here, the group where it increased vaccine uptake the most and where we also saw a significant effect modification were the most vaccine hesitant. It was those that hadn't received the vaccine in the previous season. In that group, we increased vaccine uptake close to 2.5 percentage points. As mentioned, now we've identified 2 positive notching strategies. We then followed them in the registries for a longer period of time to assess whether this increase in vaccine uptake could be translated into improved clinical outcomes. But as expected, when you increase vaccine uptake with less than 1 percentage point, it had absolutely no effect on any of the pre-specified clinical outcomes that we assessed. But interestingly, because we saw this effect modification, that those that hadn't received a vaccine in the previous season, we increased vaccine uptake with 2.5 percentage point. We also assessed this subgroup specifically. And as you can see, we saw a trend for a reduction in clinical outcomes for this specific group with the cardiovascular gain notching. And for one of them, I know this is just just picking one, and it's not the right way to do it. But bear in mind that we, in this group, saw that we actually lowered the risk of being hospitalized for heart failure and cardiovascular death, because it's gonna make sense for the next study that I'm gonna show you. So, but if a notching strategy within vaccine uptake needs to be implemented, it needs to be useful year after year. And that's why we conducted the Notch Flu 2 trial to test, does it make sense to use these notching strategies year after year? Do we still see an increased uptake? Where we just kept the useful or the positive notching strategies and tested it the upcoming year. And here you can see we randomized again the same close to 1 million participants, but used only the notching strategy that we believed were positive. And here you can see the main results of the Notch Flu 2 trial. We saw that, again, the repeat letter and the cardiovascular gain letter remained the most effective notching strategies. And most importantly, we saw in the bottom of this forest plot, you can see that we didn't find an effect modification or based on whether people were notched in previous season. So it means that it is useful to keep on notching the Danish citizens. And again, we saw that it was those who hadn't received a vaccine in the previous season where we saw that they gained the most of being notched. So it's the most vaccine hesitant that we can get out of the sofa and go and get a flu vaccine. So what has been implemented since then? The Danish health authorities have taken one of our notching strategy and now is using it on a nationwide level. They're sending a letter when the flu vaccine season starts, and then they send a reminder. They didn't take the most effective notching strategy, the cardiovascular gain letter, which potentially could evoke a little bit of fear, saying that you need to get vaccinated, otherwise you get an acute myocardial infarction or die of heart failure, right? But they at least implemented some of the strategy. So now they are notching the elderly population, 65 years and above. But in Denmark, we see that those who don't get vaccinated, it's especially the younger population who have a chronic condition. There you can see the vaccine uptake is extremely low. It's close to 20%, right? So there we really potentially can gain something from notching those who are between 18 and 65 who have a chronic kidney disease or chronic disease that also make them eligible for a free flu vaccine in our system. That's why we designed the Notch Flu Chronic trial. So here we wanted to notch all Danish citizens 18 years to 65 with a chronic condition using the same effective notching strategies that we had identified in the Notch Flu trial. And here you can see the study flow. We randomized these 300,000 Danish citizens to these different nudging strategies. And again, we saw that the two most effective nudging strategies was the repeat letter and the cardiovascular gain letter. But here you can also see that the magnitude of effect is much, much higher. The most effective nudge strategy increased vaccine uptake with close to 14 percentage points. This is really moving the vaccine uptake in Denmark, and in Denmark is a small country, but just among these 300,000, this trial meant that 25,000 more got vaccinated among 300,000. So that's a very big jump. And just to show you, we followed these participants for clinical outcomes. Very interestingly, we saw that here, where we increased vaccine uptake with close to 14%, it was the heart failure event, again, that was lowered. This is just hypothesis generating. We didn't power the trial for this outcome, but it's strange that twice now in two different populations, when you increase vaccine uptake, it's heart failure hospitalization that you potentially prevent. Very, very strange, but at least found now in two independent trial populations where we really have moved the needle. This is just to show you in the concept paper all the different nudging trials that we already are conducting. And if anyone have good ideas for nudging trials, let's collaborate and execute in this system. And just to show you that you can use nudging trials in any kind of way. In the EU Parliament elections, some of the people responsible for getting the voters to vote, they copy-pasted our strategy and did a nudging trial, testing, which kind of information letter do you need to send out to increase the voting mode. So again, you can use this concept of randomizing to different letters in a lot of different topics. Last example, and sorry for my long monologue, is this concept where we're using our registries to test the timing of drug administration. So we have coined this the circadian chronotherapy concept, and the reason why we do it is that almost all physiological measures, display this circadian variation, and the risk of events vary over the course of day. And one example is just a higher risk of getting myocardial infarction and sudden cardiac death in the morning hours. And this simple intervention of changing just when you take your drug may improve clinical outcomes, but such studies are very difficult to perform using our traditional setup because the estimated relative risk reduction is very small, right? So they need to be very, very big. And this is just to show you how we do it. We use our prescription registry to identify who gets a prescription with aspirin, with statin, with beta blockers, where we want to test whether it makes sense to take it in the evening before you go to bed or taking it in the morning time. And there we just identify them, send an e-box asking, are you willing to participate in a trial where you take your medication in the morning or in the evening? This doesn't even require informed consent because we're not changing their medication. It's just the timing of when they're taking it, and then we follow them through the registries for clinical outcomes. The first of our trials is the C3 trial, but we have several planned already. And again, if anyone have a bright idea about a medication where this might physiologically make sense, please let's execute the trial. And here I'm just showing you the C3 trial that we initiated in January last year. As you can see, we managed to randomize a lot of people, and this is just based on how often we send out batches of e-box letters. We could randomize much faster in this trial because we're not seeing them. They're just saying, yes, I want to be randomized, and then they get a new letter saying that you have to take your medication in the evening or in the morning. Okay, so now we're almost done. It's just a summary slides of the major benefits and limitations of doing trials in these systems. So the major benefits, of course, the lower cost. As you can see, we do multiple trials, large-scale outcome trials at the same time because they're not that costly. We can do many at the same time. We can recruit patients in a faster way. Using this system, we get a potentially more representative patient population, right? We at least can assess the representativeness very easily because we also have the data on everyone that didn't participate in our trials. It's a lower staff burden because we don't really need any staff when doing these trials. Rohan asked me how many people are involved in doing your 100,000 participant vaccine trials. Normally it's 3 people that is involved in executing that trial, right? Of course, we outsource the administration of the vaccine to a company, but my group is 3 people that run a trial of 332,000. It has lower patient burden. I think this is one of the most important things, which is leading to the high recruitment rate. We are not asking anything for the participants. We're saying you need to go down and get your vaccine or take your medication as you do, and then we just follow everyone through the registries. We can assess additional subsidiary outcomes. We can assess long-term effects of treatment. The limitations is that it's of course limited to whatever you have within your registries. It's less flexible for outcome ascertainment, again, limited to what we have in the registries. There's sometimes uncertainty about data quality, as we discussed earlier about the diagnosis codes, the difference between diagnosis code and adjudicated outcomes. But I want to say that in most of the large-scale outcome trials where they have both, they have assessed this difference in adjudicated outcome versus registry outcome, and they almost always show the same relative risk reduction. The absolute event rates are of course different, but the relative risk reduction, which is what we are powering our trials for, is almost always the same. It's more difficult for non-approved drugs. The safety surveillance has to be more cumbersome. There are some difficulties with multi-country studies, which I hope that we together will figure out so we can do these things multi-country. And then there are regulatory and payer uncertainty. So just the last slide, taking you through what we have gone through, the case examples that I've shown you. We can use nationwide registries in combination with this e-box system to identify and screen and invite trial participants for trials conducted using completely conventional trial conducts. That was the DECODE trial and the Coherent trial, just boosting recruitment in normally conducted or conventional conducted trials. We can use it in combination, the registry and e-box for trials conduct, leveraging the registries for all parts of the trial execution. That's our large-scale outcome trials, the vaccine trials, the Danlogic trial, the Danutrio trial. We can use our registries in combination with the e-box system as the sole intervention, both to conduct implementation trials, that's the notching trials, and we're doing many more than just notching flu. We can use the nationwide registries in combination with the e-box to test the efficacy and safety related to the time of delivery of chronically administered drugs. So this was all the case examples that we've gone through, and I don't have anything more to add than thank you so much for the attention and for keeping me awake during my monologue.
Well, I'll take the first question. That was absolutely mind blowing, I would say. I really just want to thank you for spending the time with us after the ACC and sharing this. And there's a lot to unpack and hopefully a lot of opportunities for collaboration. I do think the e-box system introduces some challenges within our system that you don't have to work through. And I guess one of them is you've heard of HIPAA, and I think that may be a limitation. On the flip side, we've talked about, they use Epic. By the way, for the fellows, this is the most accomplished fellow that I've ever met, who's finishing his clinical fellowship just now and has been training in clinical fellowship as he's been a professor and doing all this other work, which is really remarkable. So, but the My Chart in Epic, and I don't know if you have that functionality in your Epic, is a mechanism where we employ an opportunity to connect with patients and ask them whether they're willing to interact through that system to receive their information or to participate in the clinical trials. We've done that successfully in a small scale for clinical trials on this system. Could you give me your sense of whether My Chart would be a mechanism that we could employ to scale in a similar way that you've done e-box? That's question number one. And the second question I have is, I have my partner here who is great that it was here, Francine, on the hospital side and health system side. And obviously, when we're making decisions about how to implement either strategies of care or even medications that might be difficult to provide, there's a value base and a cost basis for that. And have you done any analyses of cost variation that to supplement some of your work through this system? Those are the two questions I have.
No, that's a great question. Just to start with that because remember, these systems that we use are the same systems that the government, the state, uses to assess cost. It's the administrative registries, right? So we haven't done it yet, but it is part of all the trials we do, also our implementation trials. What is the additional cost to nudge people to get vaccinated? It's great to see the numbers, right? But if you don't really prevent clinical outcomes or contacts to the TPs, then it might not make sense to use that money on, was it 25,000 additional vaccine in that group, right? So that is part of the trial assessment. It takes a longer time for us to extract that specific data source, but yes, definitely that is one of the big purposes of doing these trials. So it is part of it, but we haven't published the first of these cost effectiveness analysis, but they are pre-specified as a specific target. Also because, as mentioned, you can see that the Danish government have already implemented one of our strategies that was amongst the elderly. Of course, there we only increased vaccine uptake with 1 percentage point, so it's much less of an effect. But before we implement stuff, we also need to know whether it matters. And again, it wouldn't matter to increase flu vaccines if we don't prevent sick days away from work or hospitalization, clinical outcomes, right? So that is definitely an extremely important part of these studies that we do. For the first question, Eric, I agree completely with you that is definitely a unique source to similar to the e-box system. The only difference is that, of course, the e-box system is linked to the nationwide registries, right? So you are limited to the group of patients that you see or have a contact, or I don't know how, but you're limited to that group that you can contact. But yes, in the earlier discussions we had today about how to potentially to implement these systems, because I believe it is possible to implement a lot of the things that we do. It's just we just need to push the regulators, the IRB, this way of getting informed consent that we are getting with a video. It's, when I presented abroad, many of the ethical committee, because I participated in the ethical committee conference, because the Danish ethical committee is so proud of this, because we argue that it's much more ethical. You are opening the possibility for people who normally can't participate in trial by doing it this system, because you're randomizing them at home. And we would never ever be able to do many of these trials, the Dan flu, the Dan RSV, if we needed to get informed consent in person from 300,000 people, right? So I think these arguments and pushing the borders and pushing these decision makers to assist us, the academics, in getting randomized level of evidence in giga samples, which haven't really been something that has been possible in conventional way of doing it. If we need to, do it with all the conventional concepts of informed consent and all of this. So yes, definitely, I agree completely that is very similar to the e-box system, with of course the caveats. We just need to push this infrastructure all around the world.
Thank you. Astounding stuff. Thank you for telling us those stories. I have a little bit of a funny question, which is sort of a cultural question, and that is that how important do you think the national government directed registry culture of Denmark plays a role in, people responding to nudges and people agreeing to participate in clinical trials? I mean, we have the opposite here. People are suspicious about investigators wanting to do clinical science. And I suspect the culture is very different there. And I think, on an individual basis, by people doing individual studies here, that can be overcome to some extent, but at a kind of a mass level, it's very difficult. It's a funny question, but I wonder if you could comment.
No, I think it's extremely important, and I think as far in the discussions that I've had with many of my US colleagues about this, I think surveys in Denmark at least have shown that we trust in our authorities, and we there's also statistics showing that corruption and stuff, we don't really think in those directions when we see stuff from health authorities. And remember, the letters, it's my name often, and it's just physician, professor, and I think that is, of course, very important, that this trust in authorities, trust in researchers, trust in academics exists, and it seems as if that the current situation, even the political situation here, is pushing the other direction, right? And I agree. I believe it's, but I don't know it because we haven't done trial in that, seeing the causes. But I guess you're right that is a very, very important thing, and we at least have statistics and surveys showing that the citizens trust the tax system. They believe that. Remember, we are a country that has the second highest tax in the world, but we're all paying heavily because we believe that no one is just. But here, half of the population at least is thinking that all that money is just waste and fraudulent and whatever, and it doesn't really help, of course, that the political situation is pushing that story. So I believe it's important, but I don't know. But that is what I agree that is probably very important.
I might ask the next question. So, for trials that are lower risk, I think this non-specificity of efficacy and safety outcomes works very well, and the scale is really remarkable. But what happens when you are testing an intervention that is higher risk, like a therapeutic of some sort, where it's untested, like a new heart failure drug? How would you need to adapt the system to work for that?
Yeah, so that's a great question, and that has also been the discussion with the ethical committee and the Danish Medicines Agency, so the Danish FDA. And the short answer is you're not allowed to do this. If it's a completely new trial or a completely new drug, but one of the scenarios that I'm thinking that could potentially solve the solution is to do a phase 3 trial where you focus a small phase 3 trial where you don't power it for efficacy, but it's safety. Making sure that the drugs are safe. Then you can very fast move on to these phase 4 trial where you're allowed to test them on a broader group of individuals. That could be one solution. The other one is trying to add, more cumbersome safety surveillance, blood samples, site visits as we do. But then again, then the cost then you're not winning as much on these pragmatic way of doing trials compared to the conventional way of doing it, right? But currently, you're only allowed to use many of these concepts, the informed consent through video, if it's deemed as a low-risk trial. So you're completely right. As soon as you move into high-risk research themes, then you can't probably leverage the system in this way.
I have a question online from Dr. Erica Spatz, who also does a lot of large-scale community-facing pragmatic research. And her talk is about video consent and how do you promote representation of low literacy population, a low literacy population, like in your consent?
Yeah. That's a great question. The informed consent process is, of course, on a video, right? So the information sheet, I'm just reading it out loud. So you could potentially participate in a trial without being able to read the informed consent, but I'm not really certain whether I don't have any numbers on that, but as mentioned, in the Danflu-1 trial, we randomized 1/3 of all those that we invited. It's just 1/3 of those who received the e-box. In Danflu-2, where we have randomized, in that trial, we have randomized 1/4 of the Danish population, right? So it seems as if we are reaching a more generalizable population, but it's true. We need more randomized evidence showing that actually that it's true that we can that we recruit these kind of people, but it's exactly the argument that we at least are using, and that's also why I talked with Eric about the Transform trial, that getting data out that these pragmatic trials potentially increase the likelihood of underrepresented populations in these trials are extremely important, because again, this is the evidence we need to show those that have to take the ultimate decision of whether we can use these way of getting informed consent here as well. And you mentioned, Ron, that you have a trial where it's just where they can read it themselves and then sign. That's even more pragmatic than ours, you know. But I think we just need the evidence to show that we actually increase the likelihood of underrepresented people participate in these trials, and you know, to push this whole agenda of pushing the whole because there's so many things within the conduct of trials that just doesn't make sense, that are just, you know, as the Oxford group said in the first, you know, in the commentary in the journal, the bureaucratic burden that has been imposed on conducting research and conducting randomized trials, in particular, that makes it even harder. And that's also why we get so much observational data. It's because it's so hard to conduct these trials. And during the pandemic, it led to serious, you know, problems that people were prescribing. And that's why they wrote that commentary, because we couldn't do the trials. We used real world evidence and observational data to guide treatments for COVID. And it led to so many thousand people being prescribed stuff that had no effect, but an observation study had shown that there were low risk in those who prescribed that. They're probably more healthy, you know, those who apparently go to a doctor and get prescribed that than those who doesn't, right? So there's we just need to limit the bureaucratic burden on these kind of studies.
That is just a wonderful use of public health data that you have. And I was on a team that was trying to do with the AHA registries, and it was really difficult, you know, being a voluntary registry to really get much done in terms of the clinical trial space. And a lot of that was, you know, on the recruitment, which you talk about there, but it was also on the ascertainment. As you said, you know, some of these trials, you know, for certainly the clinical trials that we think about. So you showed some great ways of looking at it from a, you know, patient selection, and then you use the ascertainment that you have. Have you tried some other things that are kind of, and it works great with these public health, somewhat like the stepwise clinical trials of different public health through different areas of the country, because you have such great ascertainment that you can, you know, pick up much, you know, more specific data than we ever could, in terms of if you had a, you know, whether it's a public health campaign in certain areas, or even some sort of areas where you can, you know, change the healthcare, or, you know, certainly the healthcare delivery systems in certain areas over to others, because of your ascertainment is so specific.
That's a great idea. We haven't done those kind of studies in my group. I'm not familiar with if any other groups have done that. But just to be certain, you mean that we prospective randomize in a step-wedged way, or that's I think because we are able to do, you know, in many of these studies that we do, it's possible for us in an easy way to do individual randomization, right? We are doing some cluster randomized trials, but we're only going in that direction if it's completely unrealistic to do it individually randomized, right? But it's you're right. It's, you know, we can capture data sources in a very easy way, but we haven't done those kind of studies ourselves. We are planning on this season to do a flu vaccine trial where we are randomizing all Danish kindergartens to provide the flu mist. So that's a nose vaccine, just to provide it at kindergartens, but randomizing all the Danish kindergartens in the country. So we're randomizing 300,000 children between 2 and 6 years. And then the primary outcome is just whether we can increase vaccine uptake, because the children in Denmark, the vaccine uptake when they had vaccines was less than 12%. So the primary outcome is whether we can increase it to above 50%. But then we follow the parents, through these registries to assess whether the parents get positive for flu, whether they have more sick days, because that's also registered in our public system. If you're sick away from work more than 2 days, your workplace get reimbursed from the state. So we have that kind of data, and then we follow the grandparents, or whether they're being hospitalized, for clinical outcome. So because all of these vaccine programs in children for flu vaccine, they've been based on mathematical models, it's not on randomized data. So here we get, black and white evidence for whether it's cost effective to implement, these children vaccine programs, because they're said to be super spreaders. It's the kindergartens where the flu and the RSV is being spread before their grandparents are being hospitalized for these infections, right? So, but it's a great idea. But we try to do an individual randomization as long as we can, and if we can't, like this with the kindergartens, then we have to do it cluster and potentially step wedge fashion.
I have so many ideas of how to collaborate in so many ways. And I'd like to actually even formalize some of that. So I'll write you an email in the next couple days, weeks when you settle. But
That
love
sounds
to
great.
visit your group. I actually would love to invite you to join our faculty actually as an adjunct and formalize a relationship, and maybe we can learn from you. This my chart idea that I was thinking, I was like trying to think, I don't ever think anyone's tested that concept and is building on it. So just so much to think about.
That
You've
sounds-
really been inspiring.
Oh, I'm so
Have a
happy
great-
to. Thank you
please
so